thank you very much to the FSHD Society for allowing me this time to talk about Fulcrum Therapeutics preparatory studies in FSHD. These are my disclosures. And I just wanted to take a quick moment to just talk about Fulcrum Therapeutics for those of you that are not familiar with us. Fulcrum Therapeutics is using cutting-edge science to unlock gene regulation. And by placing patients and their needs at the center of our discovery and through our development engine and the associated understanding of the root cause of the neuromuscular disorders that we are evaluating, in this case it's FSHD, we have been able to accelerate drug discovery. And our focus is on in vitro patient-derived cellular models. In this case, it's the muscle, obviously. And having a clear understanding of a biomarker strategy that enables a rational and efficient path to bring therapies to patients that address the root cause of the disease. And we do this without relying on animal models. We do use them to evaluate concentration of drug in the body as well as how the drug interacts with the body. But really, we focus on these patient-derived cellular models to enable our translational medicine and also an efficient path to patients for treatment. And here's our disease pipeline. As you can see, losmapimod for the treatment of FSHD is the most advanced program, recently completing phase 2 enrollment, followed by sickle cell. And then we also have an interest in other neuromuscular diseases that are earlier in development. And like I said, I'm focusing on preparatory studies, and some of you might be asking yourself, what are preparatory studies and why do we need them? And some of you I know have participated in our preparatory studies and are participating in the ReSolve study, and we really appreciate that because usually preparatory studies do not have drug associated with them. And these studies are done so that we can answer questions about the disease, or about the biomarkers or other ways to measure progression of the disease so that we can implement what we learn in clinical trials. And by doing this, this really helps to support meaningful clinical trials so that we know how to evaluate the different clinical outcome assessments that we're looking at, or utilize different biomarkers that we may be developing. So these preparatory studies are critical to the clinical development of treatments and effective treatments for diseases such as FSHD. And when we were thinking about our clinical development program of losmapimod for the treatment of FSHD, some of the key gaps that we had identified at the beginning of this program was that we didn't really have a clear understanding of the natural history of FSHD. Additionally, we needed to identify a reliable, measurable biomarker, whether it be something molecular related to the DUX4 expression, which, as we all know, that the aberrant expression of DUX4 is the underlying cause of FSHD. And so trying to evaluate that with a repeatable and stable biomarker over time, or with something like MRI. And it is really important that as you're looking at these markers of disease or trying to understand the disease, that these measures are repeatable at different sites and that you get the same measurements over time and that they're not very variable. So what we really want from these preparatory studies is not to see differences over time in these biomarkers, but stability stability of that measurement with very small variability. And we also want to look at, we also noticed that there was not a lot of responsive clinical outcome assessments to measure the progression of FSHD. I think that some of you may or may not know that the progression of muscle weakness in FSHD is 1 to 4% over a year, and that the progression to a wheelchair is about 6 years. And so it's too long to do a clinical trial for 6 years, and the 1 to 4% progression in muscle weakness can be difficult to measure. And frankly, the clinical outcome assessments that were available to us when we started this program were really too gross to even pick that up in a reasonable amount of time. And so what we did was that we sponsored several preparatory studies and conducted them ourselves and have completed 2 of them, one being our preparatory longitudinal biomarker study, where we were looking at different ways to measure disease progression, as well as learning from you, patients like you and your families, about what's important to patients with FSHD. What should we be measuring? What should we be looking at? Because you know what is important to you and how this disease affects you every day. So we gave you our protocol to comment on it, and you, some of you might have completed the survey to inform us to make sure that we are designing our clinical trials, our critical clinical trials, in order to address your needs. Additionally, we are supporting the natural history study, the ReSolve study, which is being conducted at University of Rochester, or led by University of Rochester and at KU. And we also did an additional study looking at something called the FSHD Timed Up and Go. And I'll talk to you about that in just a moment and elaborate on that. And one of the things that we did for these preparatory studies is that we made sure that the inclusion criteria across the studies was reflective of that population that we were planning to enroll in our clinical trials. And the reason why we wanted to look at these biomarkers or these clinical outcome assessments in this population is because we wanted to be able to apply what we learned from the preparatory studies to our clinical trials, and getting a homogeneous look at a specific population in order to test the efficacy of losmapimod to treat FSHD. And so the inclusion criteria in our preparatory studies were 18 to 65, genetic confirmation of FSHD1 with 1 to 9 repeats, a Ritchie score of 2 to 4, which is a clinical severity score on a scale of 0 to 5, 0 being not very affected to 5 being affected, and is scored by the worst presenting symptom at the clinic at that time. And also evaluating for MRI eligible muscles as determined by a central reader, so that we can look at the biomarkers that we want to look at. We can make sure that we're looking at muscle, and we can make sure that we are looking at muscles that are expressing the biomarker, specifically that being DUX4. And so when we did the longitudinal biomarker study, our goal was really to determine suitable measurements that can inform the efficacy of losmapimod treatment in FSHD patients. And so we wanted to identify a molecular panel of DUX4 regulated gene transcripts, really a DUX4 signature, so that we can measure DUX4 activation, or how DUX4 is expressed in a muscle, from patients with FSHD, and take a look at that expression over time. And like I said, the goal here was really not to, was what we really wanted to see was stability here, and to look at those genes that were associated with DUX4 expression that were most stable, so that when we look at this DUX4 signature in our clinical trials, we know that the change that we see in this DUX4 signature is related to the treatment and not necessarily to the measure itself. Additionally, we wanted to establish a whole body MRI protocol, and this has really not been done before. But as you know, FSHD affects multiple areas of the body, and we really wanted to be able to capture that with the MRI and measure the progression over time. And again, with this protocol, we wanted to be sure that it can be done at multiple sites over time, and that the measurement was not necessarily variable, so that we can use it in our clinical trials, so that when we see changes in the measurements, we know that that change in the measure is due to the treatment and not necessarily to the measure itself. And then we also looked at several clinical outcome assessments to look for sensitivity of change, again, not looking again for variability, but to see if these clinical outcome assessments can be completed by people with FSHD, and if it's reliable across sites. In this study, we had 6 sites participating, and if you get different measures for each of these measurements, it can be difficult to know whether the changes that you see in your clinical trial are actually related to the measure, or are they related to your treatment. And so again, here we're looking for stability across the measurements. And the trial design is very simple. At visit 1, we assessed eligibility, and you had your first MRI scan, and we looked for a muscle that was eligible for biopsy. At your second visit, you would have your biopsy, and then 6 weeks later, you would come back for another biopsy, and we would also then perform the clinical outcome assessments and a final MRI scan. And when we were defining those genes that were associated with DUX4, so defining this DUX4 signature, in order for us to do that, we really had to be very choosy about which muscles we biopsied, because not all muscles express DUX4 at the same time. However, we know from our colleagues that those muscles that show a STIR-positive signal on the MRI, and it looks here, you can see in the circle here that it looks bright on this STIR sequence, we know that those muscles that are expressed, that are showing STIR positivity, are more likely to be expressing DUX4. Additionally, we had to be sure that the muscle had the appropriate fat content, because one of our concerns was that we wanted to be sure that we were analyzing muscle and not fat. And the picture below is something a situation you may all be familiar with, is what a muscle biopsy looks like. And once we analyzed this muscle tissue, we made sure that it had to fulfill certain criteria in order to be considered to be part of the DUX4 signature. And if the gene or transcript fulfilled this criteria, we considered it to be part of this DUX4 signature that we would be measuring later on in our clinical trials. Additionally, we wanted to develop a whole body MRI protocol in order to quantitate lean muscle volume and fat fraction in the whole body, not just looking at a specific region, but in the whole body, from singular muscles to specific areas of muscles or groups of muscles to the whole body. And here on the right, you can see the spinning man is actually an average of the fat fraction that we see in the muscles in this biomarker study. And those muscles that are green are less affected, while those muscles that are red tend to be more affected. And we will take a look at the muscle volume, the measurements of the muscle volume and muscle fat over time via this method. And the idea behind lean muscle volume is that we are measuring the amount of functional muscle tissue, while muscle fat infiltration is the relative amount of fat within that muscle tissue, and muscle fat fraction is the relative amount of fat within the muscle fascicle itself. We studied over 18 muscles and regions, and we were able to successfully show that we could image these muscles from a singular muscle to the group of muscles to the whole body, which is pictured here, and color-code it for each subject as well as for the population that was studied. Additionally, we evaluated novel outcome assessments such as the Reachable workspace, which evaluates upper arm and shoulder function. And you can see here on the left is a woman sitting in a chair and is facing the Microsoft Kinect device. Some of you might have used this previously when you played video games, and it basically captures the movement of her arm and calculates the area over which her arm moves. On the picture on the right, you see this FSHD timed up and go. Some of you may be familiar with the timed up and go, where you start by sitting in a chair, you stand up, you walk 3 meters, turn around, walk 3 meters back. However, we also wanted to make this measure a bit more sensitive and specific for FSHD by adding the component of starting from a laying down position so that we can capture some of the truncal weakness that is reported by patients with FSHD. So the FSHD timed up and go evaluates not only lower extremity function and gait, but also adds this trunkal assessment as well. And in this biomarker study, we were able to enroll 17 subjects, age 49 on average. The more males were enrolled than females. You can see here that the average Ritchie score was 3.0, but we did cover the range of clinical severity of FSHD. And through our analysis of muscle tissue, we were able to identify 7 genes that were specifically associated with DUX4, and we call this our DUX4 signature. And we were able to look at how this signature is expressed over time. So if you look at here on the right at the panels, on the x-axis, if you will, you can see the biopsy time 1 being baseline and time 2 being the follow-up biopsy. On the y-axis is just a quantification of how much that transcript is expressed. And each line here is a different gene, one of 7 genes that we are looking at that defines our DUX4 signature. On the left is the biomarker study. In the middle is our colleague Seattle Wellstone study. And then on the right is the expression of these genes in healthy individuals that came from the Seattle Wellstone study. Not surprisingly, you do not see many DUX4 genes being expressed in the healthy individuals. However, what you do see here is that when you look at the biomarker study, the Fulcrum 002-2018 and the Seattle Wellstone study, is that there is relative stability of this DUX4 signature from time 1 to time 2, and that there is significant overlap between our biomarker study and the Seattle Wellstone study. And this gives us confidence that the genes that we've identified for the DUX4 signature are the genes that are associated with DUX4, because we looked at this in 2 different studies. As well, we see stability of this measure over. So that when we employ the DUX4 signature in our ReDUX4 clinical trial, for changes that we are going to see in the DUX4 signature are most likely due to treatment rather than the variability of the signature over time. Additionally, we were able to show that the MRI protocol, the whole body MRI protocol, showed excellent repeatability over the 6 sites and over the 2 time points that were performed for those subjects that participated in the study, and that this repeatability was shown in our measurements of muscle fat fraction, muscle fat infiltration, and lean muscle volume over 6 weeks. Additionally, we found that there was an association across FSHD that showed that these measurements correlated with the severity of the disease in that FSHD patient at that time. And so you can see how the change occurs in the lean muscle volume, the muscle fat infiltration, and the muscle fat fraction, with those that are less affected or have a lower Ritchie score have more lean muscle volume, less muscle fat infiltration, and less muscle fat fraction, to those that are more severely affected have less lean muscle volume with more fat infiltration and more muscle fat fraction. Additionally, in our analysis of the Reachable workspace, which I should have mentioned previously is done with and without wrist weights, we looked at the total area that was recorded in time 1 and in time 2. And what we saw between the 2 recordings, whether the Reachable workspace was done with or without wrist weights, is that there was very little change from baseline, again, suggesting that this measurement is very stable across patients individually and also across sites. And we see the same thing here in the TUG as well as in the FSHD TUG, again, looking at the fact that there is very little change from baseline, showing that these are very stable measures of patients with FSHD. And so from our biomarker study, we can say that we identified a set of DUX4 regulated gene transcripts that provides a stable pharmacodynamic biomarker to measure disease activity and treatment effect on the root cause of FSHD, and that this is being employed currently in ongoing losmapimod FSHD clinical trials. We also developed a whole body MRI protocol and analysis algorithms to measure the replacement of skeletal muscle tissue by fat. And we demonstrated the reliability of the Reachable workspace and FSHD timed up and go assessment from time point 1 to time point 2. We further showed that the FSHD TUG is a stable measure and also differentiates FSHD patients from healthy volunteers in another study, which we collaborated with our colleagues at the University of California in Irvine with Jay Hahn and his group, where we looked at the FSHD tug at baseline and 6 to 12 months later. Now remember, our biomarker study was 6 weeks later. This is now looking at the measure 6 to 12 months later. And you can see here that the enrolled data set is essentially the same as our data set from the biomarker study. And here, when you look at the total time, the top row here is the different segments of the FSHD tug and the time that it takes to do that segment and the total time. And you can see here that FSHD individuals certainly do take a longer amount of time to complete the FSHD tug than healthy individuals. So we're able to discriminate based on the FSHD tug between FSHD individuals and healthy controls. And additionally, the timing of the FSHD tug did correlate with the severity, with our clinical severity scores here. So on the x-axis, you can see the clinical severity scores. On the y-axis, you see the total tug time. And as the score worsens, you can see that there is certainly a correlation with the severity score that's being represented here on the x-axis. And so from this study, again, this adds to the data that the FSHD tug displays excellent reliability and also is associated with the clinical severity score, that it differentiates between the healthy volunteers and FSHD subjects. And at 6 to 12 months post-baseline, there's no indication of significant change, which is great because that shows us that there's little variability even at 6 to 12 months after baseline. Additionally, we wanted to obtain your feedback on our clinical trials, and in order to do this, we did something called the virtual clinical trial. Some of you may have filled out this survey, and the purpose really was to obtain patient assessment of the feasibility of our proposed study design and also assess your willingness to participate in this clinical trial. And so we were able to enroll 79 FSHD patients throughout the world, and each subject answered approximately 30 questions about the study design, time commitment, travel, as well as clinical research. And what you confirmed for us through this study was that the schedule of assessments for the ReDUX4 trial was understandable, that the clinical outcome assessments seemed reasonable and can be completed, and that the visit duration, number of biopsies, and number of MRIs was acceptable. And hence, we made changes based on your suggestions here to our protocol, implemented them, and believe that we have the best protocol possible to answer critical questions about losmapimod's treatment for FSHD. So as I said, these preparatory studies all support our phase 2 studies, our clinical trials, and inform us about analyses and also about implementing specific biomarkers and clinical outcome assessments. And some of you may be familiar with ReDUX4 and our open label extension. And here you're going to see here again, our primary endpoint is the efficacy of losmapimod in inhibiting or reducing the expression of DUX4 as measured by that DUX4 signature that we identified in our biomarker study. And as you can see, the importance of that biomarker study, because this is our primary endpoint for our phase 2 study, as well as looking at the change in MRI measurements and those clinical outcome assessments that we previously mentioned. I think you're not surprised to see here that the main inclusion criteria are essentially the same. We are conducting the ReDUX4 clinical trial at 17 sites throughout the world, 12 in the United States, 3 in the EU, and 2 in Canada. And the study design for the ReDUX4 protocol has recently been amended due to the COVID-19 pandemic. And this was really to ensure that we are able to continue patient participation in the trial and for us to be able to monitor safety as we collect essential data to advance this important study. The main change to the ReDUX4 protocol is the increase in the study duration and the timing of the biopsy, still being conducted in 2 parts, again, just increasing the duration of the randomized controlled portion of the study from 24 to 48 weeks, but patients still have the ability to enroll in the open label extension. There are 2 muscle biopsies that are being performed at baseline and 1 at 16 weeks or at 36 weeks if the muscle biopsy could not be performed at 16 weeks due to COVID-19. And in instances where subjects could not visit the site due to COVID-19, they underwent virtual visits with the site either by telephone or by video, and safety labs were collected by home health services or standing labs or at sites if they remained open. And again, the OLE is still an option for patients who have participated in this study. So currently, we have 80 FSHD patients enrolled in the ReDUX4 study. Again, 17 sites are participating. And as you now are very well acquainted with, our primary endpoint is the DUX4 signature or activation reduction as our primary endpoint. But also importantly, our other endpoints include the MRI and those clinical outcomes that we looked at so carefully, that being the Reachable workspace and the FSHD tug, as well as other patient-reported outcomes. Our open label study is still ongoing. This is a single site study that's being conducted in the Netherlands, where we have enrolled 14 subjects, and we're looking at the long-term safety and tolerability of losmapimod in subjects with FSHD, but we're also learning a lot from this open label study. And what we mean by an open label study is that everybody enrolled in this study is on drugs. And so because this is an open label study, we have the ability to learn from the experience of these subjects in the study. We can learn about their DUX4 activity in skeletal muscle, as well as look at MRIs, clinical outcome assessments, and patient-reported outcomes. As well, we are also doing real-world assessments with mobility and wearables and looking at ventilatory functions and other exploratory markers. So I'd like to take a moment to thank everybody, those on the line who have participated and are healthy volunteers for participating in these studies. As you know, we cannot do these studies without you and without your support, as well as our management team, our clinical and scientific advisors, and other collaborators, our patient groups, the FSHD Society, and other collaborating organizations. So thank you very much for taking the time to listen to me today, and I'm happy to take any questions.
Great, thank you so much. So we have, people can start typing in their questions in the Q&A window. And for those who are listening on Facebook, we have someone there who will monitor and feed the questions over here too. So first question is, from Anonymous, can you estimate your success in the trial, or is it too early?
June, do you want me to show these questions here, or do you want to just, because I don't know if I'm still sharing. I'm not still sharing, sorry. Apologies. In terms of the successes thus far with our phase 2 studies, I can say that we have successfully recruited, fully recruited the studies, 80 subjects in ReDUX4 and 14 subjects in the OLS study. People have been very compliant, and we are grateful for that with the protocols, which is wonderful. Obviously, COVID-19 has thrown a bit of a curveball our way, but I think that with the changes that we've made in the study, I still think that we can evaluate efficacy and evaluate the potential efficacy of losmapimod for the treatment of FSHD.
Great, thank you. A couple of people are asking if there are plans for a phase 3 study.
So I can't comment on that right now. Obviously, this really depends on what we learn from our phase 2 studies. Obviously, we've learned a lot in our preparatory studies, and I think that in terms of our phase 2 studies, once we get that information, we'll be able to think about further a phase 3 study. That being said, you always hope and plan for success. So I am hoping that based on the data that we will learn from our phase 2 studies, that we will be able to support a phase 3 study and move forward with the program.
Great, thank you. And I guess the point there for people who may not be familiar with the drug development process is that the FDA weighs in at these milestones, like the end of phase 2. You have to present your data to them and make a case. Is that correct?
That's correct, June. So you have to work very closely with the FDA, especially when you're going into phase 3. In fact, they need to take a look at your program and the trials that you're proposing, and you sort of you have to be, well, for all clinical trials, you have to be given the go-ahead. But in terms of phase 3, you show them everything beforehand, and you agree with them beforehand what the appropriate trial design is. So yes, they're very involved, and not only is the FDA involved, but other regulatory agencies are involved as well.
All right, thank you. There's a question, says, could a new patient enroll in the open label study if it opens up in Kansas City, for example? I think the open label study is only in Europe currently, is that correct?
That's correct. So the open label study is only being conducted at one site in Europe. It is fully enrolled currently. And the open label extension associated with the ReDUX4 study is only open to those that participated in ReDUX4.
Thank you so much. And this is a question, I'm not sure if, I'll ask for some clarification. It's asking about the muscle biopsy requirement. I guess they're just asking, is it required to participate in this trial?
Yeah, so for the ReDUX4 study and for the open label study, it was required. The muscle biopsies were required. 2 muscle biopsies were required in ReDUX4 as well as in the open label study.
Great, thank you. We have a question. This might be from someone located in the UK. Is there a chance that there would be a study, I suppose, a clinical trial there?
So we would love to partner with our partners in the UK. Their Newcastle is a big center, as I understand it, for FSHD, and so yes, we would love to partner with them in the future. And so so if we are able to partner with them, we will definitely we will try to bring a trial to the UK as well.
Thanks so much.
In the future.
Great. Here's a question about how much does losmapimod actually reduce DUX4 expression? Is it completely inhibited? And if not, how much reduction is needed to be beneficial? This
is a great question that I think all of the experts are wrestling with currently. I don't think anybody really knows how much you need to reduce DUX4 in order to have an impact on the disease process. That being said, some experts say that any reduction in DUX4 is meaningful. And I really think that at this point, I can't give you an exact answer. I think that the field is still working on this and trying to understand it. And hopefully through these clinical trials and through the development of better animal models, we'll be able to answer that question more fully. But at this time, I don't think that anybody really knows what the right answer to that question is.
Okay, here's a question regarding the impact of exercise on the biomarkers and MRI changes that you're looking at. So I think this individual is saying resting measurements don't change much over time, and that it would require a very long study to see change in those baseline biomarker and MRI levels. But I guess the question goes to, you know, how does exercise impact those markers? Has anybody looked at that?
Yeah, so there has been one clinical trial that was done that did show that exercise improved functionality in FSHD patients. So, but how much exercise or whether exercise is going to impact our measures for our clinical trials is not necessarily known at this time. And in our studies, we said we limited exercise, especially related to the muscle biopsy, because certainly exercise can affect your results of the muscle biopsy. And we asked that people continue with their usual routine so that we can minimize the potential impact of exercise on us getting an accurate readout for the efficacy of losmapimod to treat FSHD.
Okay. There was a question about the timed up and go task. Are subjects getting up from, are they allowed to rise from lying down by pulling on pant legs or other techniques to get out of bed, or do they have to be completely like unassisted?
Yep, so we do allow assisted devices in both the FSHD tug and the tug. So assisted devices are allowed, and if you need to pull up, that is allowed.
Okay. There's a question, it says, you mentioned something about the progression of FSHD. Is there enough time between the measurement periods in the trials to understand regression and be able to project future decline? So I think to maybe try to paraphrase that, you mentioned these measurements are very stable over time. Mm-hmm. So does that make them difficult ways to measure an improvement or a decline in progression if the mm-hmm. decline is hard to see?
Yep. So with these measures, what we're looking for, again, is stability over time. But if we're thinking that we're living in the world of improvement, if the usual natural history is for people to decline with these measures, it would create even a greater difference between what people were be able to perform on drug if they're improving, and then if the natural history is either to stay the same. I wish I had a whiteboard. Sorry, everybody. Stay the same or going down. So if you have somebody that's improving and you know the natural history is to stay the same, you would see this difference. If they're improving like this, but then they're going down, you even have a larger difference. So we actually took a more conservative approach by looking for stability rather than looking at progression.
Okay. Question here. If the study is successful, what will be the effect for people with FSHD1?
So when you say that the study is successful, June, is there any?
There's not a definition. I think it's probably not mm-hmm. the researcher's definition, which is that you have great data and the data could show that the drug doesn't work, for example, but mm-hmm. you have great data and you know that that's a really reliable result. I think from a patient's perspective, it probably means the drug is working. Mm-hmm. So in your best case scenario, knowing the mechanism of how losmapimod mm-hmm. works, if it has the outcome you know that you're hoping for, what would that mean, I guess, for patients?
Yeah, so if we have the outcome that we're hoping for and we see efficacy with losmapimod treating the underlying cause of FSHD, we would move on in clinical development to a phase 3 study and try to work as quickly as we possibly can to get the drug to market and in the hands of patients so that we can start treating people as quickly as possible.
Okay. And the hope for outcome from a drug that passes through that would be that people would stop progressing or possibly even improve, or what?
Yeah, so yeah, exactly. So currently we're looking to stop progression, but improvement would be wonderful as well. And we'll learn sometime next year what world we will be living in mm-hmm. Related to the outcome of the trial.
Oh, great. Question here, when is the phase 2 anticipated to be completed?
So the anticipated completion date for the phase 2 study is Q1 of next year, early okay. Q1.
All right. A question about the open label study. Will it complete around the same time as the ReDUX4 trial? I guess the phase 2.
Yeah.
Trial.
Yeah, it'll complete around the same time.
There's a question here. How do you recruit subjects? How do you get the word out And then you mentioned in a slide that we have 1 to 3%, I guess it's change in muscle function or strength. And that 1 to 5 years is the usual progression for wheelchair use. Did I hear that correctly? A little clarification on that.
Sure. Published data shows that the progression of muscle weakness is 1 to 4% per year, and that the average time to wheelchair is about 6 years for 24% of people with FSHD. And apologies, June, I didn't. The other part of the question was.
I think that was the and what was the source, and I think that is from the National Registry, one of the studies run on the U.S. Registry. And when you say the time to progression to wheelchair, that's in a subset of patients who have a risk factor, right? They have, it's not just any patient will within 5 years progress.
No. That's the number of patients that, yes, that's correct. That's the number of patients that that can progress to wheelchair in 6 years. That's correct.
Right. In a phase 3 trial, how many people would you need to recruit?
For a phase 3 trial, it's slightly different than a phase 2 trial, because our clinical endpoint in the phase 2 trial is really looking for efficacy based on a biomarker, which is reduction of the DUX4 signature or DUX4 expression, as measured by the DUX4 signature that we've identified. In a phase 3 clinical trial, what is really required is showing clinical meaningfulness or clinical benefit, meaningful benefit on a clinical endpoint. And hence the reason why we are working so hard to find a responsive clinical measure, such as Reachable workspace or the FSHD tug, in order to allow us to assess the change over a reasonable amount of time. Like I said, FSHD is a challenging disease because it is fully progressive, but that does not necessarily mean that we can't measure other changes over time. And hence, I think that we just need to develop better clinical outcome assessments in order to capture that progression. The current clinical outcome assessments that we have right now are too gross, and we can't pick up the subtle changes in patients with FSHD. But by looking at these novel outcome measures, such as the Reachable workspace or the FSHD tug or even others, we could potentially identify a new or repurposed, in this case, the FSHD tug is more of a repurposed clinical outcome measure that can inform us of changes and clinical benefit for a phase 3 clinical trial. And depending on what that benefit is and how much change we can measure, informs how many subjects we need for a clinical trial. So if the change over time is little, you're going to need a lot of patients. If we're able to measure larger changes over time, then you need less patients. And so we will be doing thoughtful analyses to inform us based on what the clinical measure or clinical outcome assessment we choose to be our primary outcome for our phase 3 trial, and then based on how many subjects we need based off of that in order to see a difference between treatment and potentially placebo for our phase 3 clinical trial.
Mm-hmm. I just want to throw in a little advertisement there for the importance of participating in these preparatory studies like the natural history study, the Resolve study, because that's where those instruments, those measurement tools are being developed. And the more sensitive, the ideal thing is to find a tool that will let you do the phase 3 with the minimum number of patients, because every patient recruited increases the cost of the trial, the time of the trial, and so on. So
that's right.
I just needed to say that.
No, I can't stress enough. the meaningfulness of these preparatory studies. And that's what this talk is really about, is that in order for us to have successful clinical trials, we need to understand this information, and hence the reason why we spend so much time on these preparatory studies. Because in order for our clinical trials to be meaningful, and in order for us to understand what these assessments are telling us, we have to understand how things perform under what we call natural history conditions. And if we can understand that, then we can, when we have it in a clinical trial and we see changes in that measurement, in that clinical trial, we can ascribe those changes to the treatment rather than to the measure itself.
So here's a question asking, the average age of the trial patients seems high given the average onset age of FSHD. Will you be recruiting younger patients for a phase 3 trial?
So I can't comment on it because the phase 3 trial has not been designed yet. So, but I think that we will be considering all options are on the table for a phase 3 clinical trial.
Yeah. One thing I'll say is that in general, younger patients tend not to get involved with research or even with advocacy. If we look at the demographics of the FSHD Society, for example, it's a pyramid with the smallest number of patients in the teenage, young adult years, and every decade, the number of people who are involved grows, but we know that doesn't reflect the population that's affected. So that's a big part of something we're working on, is how do we increase the engagement amongst younger patients. It's not that they're not being allowed into the trials, it's just that they're not volunteering at the same rate that older patients are volunteering.
Right, and one of the things that you can do so that you can ensure that you have a representative sample of what the population looks like is that you have quotas, essentially. We call it stratification in clinical trials, but you can have quotas in which you say, okay, we need this many patients from X age to X age in order to fill those certain buckets. And that is something that could be considered in the future.
There's a question if you are close to having a non-invasive way of measuring DUX4 expression. So
yeah, that's a great. Yeah, that's a great question. And certainly in those exploratory biomarkers that we're looking at that I kind of glossed over, we are certainly looking for non-invasive ways to look at DUX4 or to measure disease activity in FSHD, which would be looking at different blood tests, different proteins in the blood, things like that. So we're working on that as well.
And the FSH Society is working on a workshop to try to accelerate the development of that type of non-invasive biomarker. If a phase 3 happens, will it be possible for patients to travel to a country that has a trial? That's probably a regulatory issue of whose, nationality and trial participants.
Yeah, and to be honest with you, it's for speaking in general, and I agree with you, June. It really depends on the way that the trial is designed and what the sponsor has set up for patients to travel and to be able to participate, going from one country to another country. And so just in general, it's really trial dependent and sponsor dependent. And I can say that from here, we have not made any decisions at Fulcrum regarding that at this time.
Great. There's a question whether there could be compassionate use allowed that goes concurrently with, I guess, with a phase 3 trial.
Yeah, so the compassionate use is being explored at Fulcrum currently, and so we will certainly be notifying the FSHD Society once that program comes online, but it will likely not come online until we have some idea of efficacy for losmapimod.
There's a question about the dosing protocol. Is 15 milligrams twice a day, is that what the dosing protocol is?
That's correct. That's correct. Yeah. And that sorry. Yeah, go ahead.
Go ahead. Go ahead.
Yeah, I was going to say that dosing for the 15 milligrams is based on our own data as well as modeling data. And looking at the PK parameters or the concentration parameters as well as PD parameters, meaning target engagement for inhibiting P38, and evaluating how the losmapimod at different doses engages and inhibits P38. And I think I had seen in that question, because I saw it pop up, why we weren't going higher. And the reason why we're not going higher is because we don't necessarily get any more target engagement from higher doses. So, and when you start going higher, of course, then you increase the potential for adverse events and things like that. So currently, based on our modeling as well as on our data, 15 milligrams is the optimal dose.
Great, thank you. There's a question here that we hear a lot. Why is age 65 the cutoff age? Many older patients may have more time to participate.
Yeah, that's a great point. Part of the reason why we chose age 65 had to do with the occurrence of comorbidities. That as people get older, they have more health problems, unfortunately. And so in order to be able to limit the number of confounding factors in our clinical trials, we had to choose a cutoff where people are still relatively healthy from, an age perspective, in that, they're at decreased risk of cardiovascular disease and things like that. That could potentially interfere with the clinical trial, and hence the reason why that upper limit of 65 was chosen. I think that we have talked a lot at the FSHD Society, the importance of, in clinical trials, homogenizing the population so that you can decrease the amount of noise, if you will, from other factors that can influence your signal. And so if you have a lot of patients who are at high risk, let's say for cardiovascular disease or something like that, you might have a lot of people that are starting to have heart attacks or strokes, not necessarily because it's related to your drug, but because it's related to underlying comorbidities or propensities to have that disease. And hence the reason why, it's very tough, and it's something that actually requires, when you're thinking about this inclusion criteria, requires a lot of discussion and thought, as well as modeling to figure out exactly what that age criterion should be.
A similar kind of question we've gotten is from people who are in wheelchairs and asking why they are excluded.
Right. So currently, we are still trying to optimize our clinical outcome assessments for those that are ambulatory, so that being the FSHD TUG, as well as the Reachable workspace. However, we still, for patients in wheelchairs, there are other clinical outcome assessments that can be applied, and we need to understand those clinical outcome assessments in the FSHD population. So, so we need to have additional preparatory studies to evaluate what something like the Reachable workspace looks like, or what other upper extremity assessments can look like in that population, in order for us to be able to employ it or put it into a clinical trial.
And then on the other end of things, you have participants who are have very mild symptoms or almost asymptomatic, but they're genetically FSHD. Do you need any of those for a trial?
Yeah, and if you are thinking about those, if you're trying to measure a difference and you have people that are not showing any signs of the disease, and they're already at what we call the ceiling for some of our measures, it can be difficult to measure the change that occurs with treatment. And so that's not to say that just because we are looking at people who have a specific clinical severity score, that this drug won't be given to people who aren't necessarily symptomatic. It just means for our studies right now, we need to enrich the population so that we can capture the signal in order to make an assessment about efficacy. And so hence the reason why we have that criterion in there of a Ritchie score of 2 to 4, because we need people who have some disability that we can measure, and that then a disability that, or a, what I should say is not necessarily a disability, but an impairment that we can measure, and then measure change in that impairment.
Right. So if the drug does get onto the market, somebody who has very mild symptoms could still take it if they have an FSHD diagnosis, and hopefully it will prevent them from progressing as fast as they might otherwise.
Yes, potentially. And then it all comes down to, again, regulatory interactions and what in the end comes out of our phase 3 clinical trial, and what's agreed upon by the regulators about who the appropriate population is. But certainly, again, nothing is off the table. Nobody right now is excluded from receiving this treatment. Right now, we're just trying to capture the signal to see if losmapimod is efficacious for the treatment of FSHD. And if it is, it will certainly be looked at, I'm sure, in multiple populations and applied.
Okay, so I think we're past the end of our time, but I have one final question that I think everybody wants to know the answer to, which is, assuming all goes well, what is the best case scenario for when the drug could be on the market?
Yeah, unfortunately, I can't answer that at this time. As you know, getting a drug to market is a really complicated process that requires lots of more collection of data, discussions with regulatory agencies, and with others that provide pricing of the drug, things like that. And so, unfortunately, I can't give a granular answer to that. We are working as hard as we can to get this, that if losmapimod, or when losmapimod shows efficacy for the treatment of FSHD, we are working very, very hard to get this to patients as quickly as possible. And please know, as I said at the beginning of my talk, we're really patient-centered. It's the patients who are our driver and who we are working for. We're working for you, and we are trying to get this to you as quickly as we possibly can.
Great, thank you. And I think I'll let comment from Mark Stone to wrap up today's webinar, which is, from my perspective, Fulcrum has been very thoughtful in your trial design. This is very important to the success of any trial. So on behalf of our FSHD community, thank you. And I'll say thank you on my own behalf too.
Well, thank you everybody for taking the time out of your busy days to listen today. And if there are any other questions, please feel free to reach out. June knows where to find us.
Great.